Welcome to episode 3 of New Horizons. Today's guest is Jeff Hsu, the co-founder and CEO of Ivy Natal. Before Ivy Natal, he led bioinformatics at a startup focused on pre-implantation genetic diagnosis. He also completed postdoctoral research at the Cleveland Clinic, where he studied the genomic basis of heart disease. Jeff earned his PhD in molecular medicine from Case Western Reserve University. I think this is going to be one of the most transformative technologies of the next 2 or 3 decades, so it's a very exciting episode. Infertility will be a thing of the past. Same-sex couples will finally be able to have children, and there are even more implications for his specific technique that I hadn't thought about until I read his blog post, which I'll link below. So I think it's going to reshape society quite a bit, and for context, this was recorded on March 8th. Please welcome Jeff Hsu. So first, if you could tell us how far along this endeavor is, what year did you start, how many employees are there now, and why did you decide to work on this?
So my company, Ivy Natal, relatively small player in the space. It started about 5 years ago in 2020. And right now we're at about 3 employees. And we're focusing on one particular issue of the broader field of in vitro gametogenesis. And that's specifically trying to induce meiosis as efficiently and starting from any arbitrary cell type.
Nice. So from what I've heard, there are several different ways to do this. The creation of germ cells, that is. The method I'm most familiar with is where they take a skin cell or a blood cell, revert it to an iPS cell, an induced pluripotent stem cell, and then they guide it to becoming either a sperm or an egg by exposing it to the necessary biochemical signals in, a petri dish, I imagine. This is different from your approach, right? You skip a lot of steps in direct meiosis induction. Explain that for us, please.
Yeah, so the common way that it's usually done is first the conversion to induced pluripotent stem cell, and then a lot of groups will turn it into what they call an iron milk, and then from there into a PGCLC, and then into the spermatogonia or oogonia. Our approach is trying to skip a lot of those steps and go directly from really any cell type, whether it be a skin cell or some other cell type. Although we are working with iPSCs right now, just in culture, lets us really do a lot more experiments for the passaging. And so, although we have tested some stuff on fibroblasts.
I know there were mice with 2 dads and 2 moms created by a China group in 2018. And then there's a group in Japan as well, led by Dr. Hayashi, that has made a lot of breakthroughs. Were those methods similar to what you're doing, or are those different?
Yeah, they're similar. They have to do more, though, for the same bipaternal maternal mice. And so one of the big things that happens during germ cell development is the germ cells undergo a pretty massive demethylation genome-wide. So DNA methylation is essentially a epigenetic mark that represses gene expression of adjacent cells. And normal somatic cells will have quite a bit of DNA methylation. When it undergoes germ cell development, it loses global DNA methylation goes down to something like 5% of what it is in a normal somatic cell. So that's quite dramatic. Once it, however, gets to sort of the male-specific germs, is it the sperm, or the female-specific oocyte egg, there are certain regions that are differentially methylated between the male and the female. And it's critical that when you try to produce an organism that takes basically both 2 females or 2 male, that you actually change those imprinted regions so that there's essentially a matching male and a female imprint, even if the original genetics comes from the a male or and so they had to basically edit those. So the groups in various ways. So there's a couple groups have now done this and have produced live mice, and either by editing the imprinted regions directly, or they actually go in and demethylate. You can actually go in and surgically demethylate or methylate the imprinted regions using a Cas9 CRISPR-based approach. And so, yeah, so that's been done, and it's about 20 regions that they had to change for like the bipaternal mice to male mice, and they're able to get live mice, but it's not like it's still at somewhat of a low percentage. But it's definitely doable. And groups have done different strategies to do that. And I think that as things get better and the technology there matures and the exact loci that you need to make changes to, you'll make that pretty, that efficiency quite a lot higher. And it's kind of interesting why you need sort of like a balanced imprinting. There's like evolutionary reasons why there's some sort of there's both like male specific and female evolutionary pressure on shaping some of those regions, and that's been known for quite a while now.
Interesting. And so do you have to deal with those imprinting issues as well with your approach?
Yeah, we'll have to deal with that as well, to make sure that when we try to create a viable organism from our process, that if we want to do the 2 male or 2 female embryo, that they have that balanced imprinting. Yeah, I think like those really for I think the field in general is matching like the epigenetic marks to result in like a viable developmental embryo is one of the key challenges, because I think we're getting to the field in itself is getting to the point where we can essentially create like an oocyte that's very very similar to what a naturally derived oocyte, and same with on the sperm side as well. And so just getting like everything tuned to the right levels, I think in a lot of ways is going to be a key challenge. In the end, it could be much harder than we think it is, but there are a lot of landmarks and a lot of guideposts to show like where we are and what we need to get to the final end product.
Yeah, I was going to ask what one of the biggest technical challenges are, but it sounds like it's imprinting. So how do you validate that the egg cells you create are functionally equivalent to natural eggs?
Yeah, that's going to be, I think the metric has been to essentially create live animals from that. And so one of the key challenges going forward is, we're doing this, a lot of groups are doing this on mice or rats or chickens, but moving to like a non-human primate is going to be the big expensive step to do, and that's going to be a big jump in itself, and then finally moving to humans, and so there's functionally, that's at the end of the day, that's what you kind of have to optimize for, is like a viable developing embryo, and watching the developmental milestones that entails is, you can get good proxies of if that will result in a live birth, but it's only going to be, at the end of the day, it's like the live birth percentages is going to be key, and eventually like full development into like adulthood. So for the mice stuff, they've shown mice that have gone on to create multiple generations after using like an in vitro gametogenesis process. So there's some amount of confidence there. But yeah, to validate the actual functionality, that's going to be pretty tough. We can match the transcriptome, we can match the epigenetics, but like, yeah, functionally, you don't really only have like being able to generate a full developmental, fully developed like embryo.
Yeah. So something I noticed is that there are a few different startups working on creating egg cells through IVG, but none working on creation of sperm. And the reasons for this are interesting. A big part of it is just the fact that sperm are more complex than eggs. Egg cells are a pretty simple type of cell structurally by comparison, so it's a harder task. Also, there's probably more demand for egg cells than sperm cells because women lose their eggs and their ability to reproduce as they get older, but men largely retain sperm for their whole life. And then also, if we're talking about the creation of eggs from male cells versus the creation of sperm cells from female cells, then there's another big factor. For males, you have XY chromosomes. So what you can do, from what I've read, is you just create another copy of the X so that you have the two X chromosomes you need. However, for the female cells, you just have two X chromosomes. So you would have to construct a Y de novo. Or I think another option would be since the Y chromosome is a lot smaller than the X, you could potentially just add the necessary Y chromosome genes onto the second X. And that would only be 10 to 50 genes, it seems. So I guess, do you think you guys would try to address that problem as well once you accomplish the creation of eggs?
Yeah, I think, I think eggs are obviously, for the reasons you mentioned, sort of the more economic sort of viable option. There are startups though that work on the sperm side. There's one called Bio, and I think that they just started last year, and they're based out of Salt Lake City. And I think there are a couple other ones as well. But yeah, I don't remember the exact details. In a lot of ways though, the egg is more challenging, I would say, because mainly, at least with IVF, there are sort of procedures where if you get the haploid, cell nucleus, the rest of it kind of doesn't, as long as it has the correct imprints, the rest of it functionally doesn't matter, because what they do is they just, they inject the sperm into the egg already in a procedure called intracytoplasmic sperm injection, or ICSI, which is common in IVF already. And so, so the egg is challenging because there's, it has, in a lot of ways, it has to recapitulate the whole developmental trajectory of a developing embryo. It has, there's a lot of maternal transcripts that it carries, and the sort of the embryo doesn't actually start transcribing until a given point in development where paternal transcripts will actually start being made. And that's called that's the ZGA, or zygotic genome activation, when it starts transcribing new mRNAs. But yeah, so the question you mentioned with the female, if you're only focusing on eggs, the interesting things the groups are trying to do the creating eggs from male starting material, and that seems to work besides the imprinting that I had mentioned. And in the case where you're trying to do let's say two females are trying to have a child and they want a male mice, and you had mentioned, where does the Y come from? I think that's going to be pretty an interesting problem. Now, obviously, there's not that many genes, as you mentioned. There's not that many genes on the Y, and so either adding on the genes is one possibility if they want a son, or de novo. I don't think we're near. There's been a lot of de novo synthesis for bacteria and stuff, and use of artificial chromosomes. I don't think they've gotten anything quite close to the Y yet. I could be wrong. That field's moving pretty fast, and as the DNA synthesis begins to continually improve year after year, that could be a possibility. But maybe adding the Y, or maybe fishing out a Y, or finding a Y chromosome donor and just using the Y there is a possibility. For one of the paternal efforts, they did get, in the course of culture, I don't actually remember the details, but they did have to get rid of one of the X's and the Y's somewhat stochastically, I think it was the Japanese group's paper. And so they had to go look for the worm that ended up missing. I know there are efforts to try to specifically digest a given chromosome. So if there's like a trisomy, to digest the extra chromosome and stuff. I don't know how far along those efforts are, but
I see. What's your time?
yeah, I don't know if I answered the question.
Yeah, no, that was good answer. What's your timeline or prediction for when this will be available? At a minimum for like women with infertility, because that's probably the application that's going to come first.
Yeah, that's man, I am very bad with timelines. Like, he asked me 5 years ago, I would have said like 5 years, but clearly that didn't happen. I think a lot of the people in the field, like the more, especially on the academic side, are saying at the very earliest, like 10 years. And there's other, some like startups, and it could be 5 years or 4 years. I don't think it'll be like within like 2 or 3 years. I don't think, just because there are still a couple like technical hurdles people have to overcome. But it could be, there are people that are pushing the technology very fast. We'll see. Yeah, it's really tough to really project that out. For us, it seems a lot longer given the size of our team and stuff. But I think for there are other efforts out there that are moving very fast and are very private of where they are. So I can't really say. They could be much further along than we are. And so, yeah, they could have a lot of data showing that they're just really, maybe a year or two away.
And then presumably it would take longer for like gay couples and transgender people and those sorts of applications.
Yeah. I don't think it'll be that much longer. I mean, just for the progress for the field has been for the epigenetic editing and printing editing. I think it'd be interesting to see with some of those tools. I feel like might be even become standardized for just like molecular biology, because there's a lot of interesting biology there. So there'll be like cocktails to modify to change like a male imprinting to like a female imprinting, just for Yeah.
Do you anticipate problems with things like ethical approval, making sure that this is accessible to people? Because I worry for people that some illiterate politician will try to ban it last minute.
Yeah, I think, I definitely think that's going to be a potentially big issue. One of the big motivating factors for like the approach we're taking is to try to make it sort of like accessible as possible, as scalable as possible. So, and then with the ethical concerns too, like I think those are always going to be pretty tough, but we can try to ameliorate some of the concerns, or at least the field can potentially ameliorate some of the concerns. Obviously, looking back, IVF is also highly controversial for its time. And so, yeah, I think the approach we're taking, and others, just ensuring like the combination of like the safety and stuff is going to be critical for like acceptance, not only on the ethical side, but on their regulatory side, whatever form that takes. I do think like infertility is obviously like a big issue, and like so a lot of people see that as a growing concern. But yeah.
Mm-hmm. And I'm sure there will always be like jurisdictions where it's fine, like different countries, different states, etc. will have different rules probably. What do you think about the more out there possibilities, like single people having children with themselves, or 90-year-olds having children and stuff like that?
Yeah, like self babies. VCs love that because they love themselves. No, I'm joking. Don't cut that out. I'm joking. I think the single people having themselves, I think, is actually pretty hard. Mainly because like humans, on average, each carry like I forget the exact number, but it's I'll say around 6 around like homozygous lethal mutations. And so you have to make sure when you're doing like the alleles that you choose genome-wide, there's none of those happening, because those, I think so. That's a hard problem, and yeah, but doable. And the older people one's kind of interesting too. I still, that's kind of an unsettled question, right? Because we're having a lot of, in a way, it's sort of like, males have always been doing it for quite a while. There's a lot of 80-year-olds already having like children. At that, that possibility hasn't quite accessible to females. Although if you use it like a donor egg, there have been, 70-year-olds that have carried, women that have carried children, but that's with a donor egg. But I think the older people having children, would this technology increase that? Probably, but we already have it in some form.
Yeah, I don't think it's like a problem. So IVG could also have interesting implications for gene editing of the embryos. Right now, if people want to choose a trait for their child, which is already possible in a very limited degree, like it's possible to choose sex and eye color, and it's even possible to select for an IQ increase of like 2 to 3 points. If people want to choose traits, right now they can only choose from among a very small number of gametes. But once we're able to do IVG, then it'll be possible to choose from a very high number, because if you're starting from things like skin cells or blood cells, then you could make like 100 or 1,000 eggs or sperm and then choose among them and have like a much larger effect.
Yeah, exactly. Just having the starting material and more starting material. Yeah, so I think there are groups now that are trying to do like, multiplex editing and then for traits. And, for us, really, it's just, creating, germ cells.
This is more like later.
Yeah. And there's, there are some scaling things, even if we create like a lot of gametes, it's, it kind of goes like the selection goes log to the number of things you're selecting for. And so, or number of individuals. And so it still does taper off a bit. But I think one of the more interesting applications of what we're trying to do is that you might be able to select at the haploid stage too, I think that's going to be critical for, adoption of this technology. Not only does it give you more power, but it also, like, if you could select at the haploid stage, you would be able to you wouldn't be creating like extra embryos. This is all like very theoretical stuff, but we can maximize the chance of like a live birth.
Wait, why is it better to select at the haploid stage?
So you can, you're selecting, you could essentially pick from the best of two instead of picking at the embryo stage where they already have combined. And in addition, there's obviously religious concerns about discarded embryos. And so if we can maximize the chance of success at the haploid stage, I think that's, in a lot of ways ideal. Yeah, but again, that's like speculative. But that's something like our technology would allow that isn't just like more N or whatever, more like things to select on.
Mm-hmm. So far we've focused on the like IVG aspect, the creation of gametes for reproduction. But there's also, this is also very relevant for rejuvenation, for de-aging. Could you explain that to the listeners?
Yeah, so this is very speculative, and we have a blog post on this. So just to preface it, like, this is going to be highly dependent on how efficient our process is, as well as essentially sequencing costs coming down quite dramatically. But the idea is that if we were to induce like meiosis in any arbitrary cell type, what we can do is, because as an individual ages, they accumulate somatic mutations, and we know actually like the number of somatic mutations is essentially inversely correlated with the lifespan of an animal. So even though like, mice and elephants differ, widely in the number of cells they have, how long, at the end of the life, the number of mutations, somatic mutations, only like differs by 3 or so across a wide variety of mammalian species. So we know that like, and like even just like from, just thinking from first principles, mutations will, potentially lead to like a whole host of issues, including like senescence of cells. And so is there a way, to essentially de-age, go to an earlier stage before these mutations have accumulated? And in a lot of ways, there is, in the sense that like different parts of your body will accumulate different mutations. And so if you want to go to an earlier stage, what you can do is, it kind of gets complicated, but like you run meiosis, and then you have like a so-called, air quotes here, breeding population of cells from different lineages. And what you can do is just repeat the process and select cells that have the least number of mutations. And so once you do our process to get these recombinant haploids, you can fuse the 2 haploids that have the least number of mutations, and then repeat the process over and over again until you get to a point where you essentially reduced the number of somatic mutations down quite dramatically. And so that's going to be dependent. Yeah, so the idea there is you don't really need to know what the mutations do. You just know, all you need to know is you want to go to an earlier germ-like, more germ-like state. Germ-like, I mean, as like an earlier ancestral state. That's sort of like, and so I think that's the appeal of this process, is that you essentially dilute out all these somatic mutations that individual has accumulated across their lifetime. And so, yeah, there's why that's kind of useful is not only like you would get essentially cells that have been de-aged, and then from there you can use them, to generate like as a source material for cell therapy. So like there's already CAR T and bone marrow ones. So if you wanted to generate like a more robust cell before you like inject them in and have them be, less prone to senescence, less prone to just apoptosis and dying, and less prone to essentially eventually maybe even transforming into like a cancer cell, then I think this de-aging process is kind of cool, and it's an interesting way to do it. And you can combine this potentially with like gene editing, because there's like known recurrent mutations that bias the cell to like grow and outcompete other cells that don't have that mutation. And so combining, just a hybrid approach will probably result in like the most robust cell type that you could use for like a cell therapeutic. And so for this to happen, there's obviously, as I mentioned, it's speculative because there's a lot of things that need to happen, including reduction like sequencing costs. And then for us, what matters too is that you need to be able to fuse 2 haploids together and have them divide. There are ways to fuse cells together, and people have been doing this, but we haven't done it with the results of our cells yet, and that's something we need to prove out after meiosis. And so, yeah, so this is like an interesting way to do that, rather than going through and sequencing, finding like the thousands of edits that you need to make and design like guide RNAs for that. This is an alternative approach.
Interesting. Yeah, I had never heard anything like it before I read the blog post. So it's kind of like breeding your own like skin cells together. You like turn them into haploid cells and then like breed them basically, and then do it iteratively over and over.
Yeah. So the iterative approach is pretty critical, because that lets you dilute way more mutations out.
Yeah, and the reason the mutations go away is basically just like natural selection, right? Because if a cell is too messed up, like too many mutations, then it's unlikely to succeed in the next round of reproduction. So like over seconds.
Yeah, exactly. I actually didn't talk about that in the blog post. But there's very interesting strategies about to do this without sequencing as well. And sequencing just makes it easier and it's like simpler to understand. But you could actually do this without sequencing just by like the evolutionary pressures you mentioned, as well as some proxy metrics for like robustness that you can essentially select on in culture.
I see. But how do you like score them? Deployed diffusions.
Yeah, you would score them by sequencing as the easiest, simplest way. But that's more expensive. That's why that's why I need sequencing costs to come down for this to work. You can just, the evolutionary way works really well too. With the exception, there are known mutations that I mentioned that essentially bias like the survival rate, like mutations in like TP53, which is a known, like the known cancer mutations and stuff.
Yeah, so that's like a weird exception where like in some cases, being very like fit and prone to survival in the in vitro case does not translate to the same thing in vivo because it translates to like a higher cancer risk when you...
Yeah, exactly. I mean, a lot of the cancer mutations are like that anyway, so, but yeah.
How long would each cycle take?
Yeah, so that's something that's also like very critical for us, with the meiosis induction, at the limit, yeast can do that at 4 hours, but they're at much smaller chromosomes. We're hoping to get this under like a week or 2 weeks. But that's still, I don't think we're there anywhere near there yet. In some of the protocols, especially from the way where the for differentiation, different the cells, that they're, for them to get to meiosis, that takes like several, 100 some odd days. Other groups have kind of got that down using different approaches. And for us, we haven't gotten full meiosis yet, but we're hoping to get to, something that's under like 2 to 3 weeks and hopefully shorter. We'll see if that's possible though.
Nice. And then how do you induce the meiosis in the first place?
Yeah, so we're basically upregulating like transcription factors. Yeah, that we think are critical for inducing meiosis, as well as some other particular genes. So we have like a cocktail of genes that we're turning on, as well as some small molecules to help that transformation happen. Yeah, so it's just like we have some like messenger RNAs that essentially lets us transfect the cells. And we just have a cocktail of them.
So in practice, would the plan be to integrate these cells back into the body? Would it be some sort of like injection? How would it work?
Yeah, so that would we'd have to, that's not our focus, but for like CAR T or autologous ones, that would be like an injection in the bone marrow. That'd be like a transplant. But we in the future, as these other companies, these other startups to like work on like potentially totally in vitro organogenesis, that's also another possibility where they just like grow a whole organ.
How do you know that it wouldn't elicit an immune response?
Yeah, so that's a really interesting question. And so in the 60s, the mice lines that we work with are completely inbred, meaning they're homozygous everywhere. And so in the 60s, there was an experiment where they bred, 2 lines together of black 6 and a BALB mouse. Inbred mouse lines, they produce something called F1 generation that are heterozygous everywhere, meaning they have one allele from each strain at every variant position, polymorphic position. And our process would essentially be breeding like the F1s together. And so recombination happens between the maternal and paternal chromosomes, and that produces like a mix. Some locations will be completely homozygous for one strain, and other locations will be homozygous for another strain, and some locations will be heterozygous. You can breed those again with each other, the progeny of an F1 mania together, to produce an F2 generation. You can do that again to produce an F3. And so that's very similar to the process that our iterative approach would be. What they did, they won on and did was take F2, F3, and F1s. They did a transplant from the F2 and F3 generations back into the F1 generation. And then they looked at the rate of rejection. So they did like 100 of these for each mouse. And they basically, they saw like one rejection in a lot of cases. And those could potentially be like somatic mutations. So that kind of follows at least like the theoretical prediction that there would be no rejections. But essentially, why that would be is essentially the product of a mating between like modulo, like the somatic mutations, which you're trying to dilute out anyways, is that they would essentially have like the same genetics, even though some would be like homozygous or at certain loci, but they would still be like homozygous for, from one of your either maternal or paternal chromosomes. And so there shouldn't be a host rejection of that. There are some complications though. You probably don't want recombination on what we call like the MHC, like some of like the immune, like the actual immune proteins, because those are there's definitely like we call like a genotype by genotype interaction there if you have a recombination in those haplotypes at the MHC complex coding regions. So this is, it's theoretically they shouldn't be rejecting but there's definitely like a chance. And so that's something we're going to have to figure out. Because basically what you want to avoid is like these genotype by genotype weird interactions. And evolutionary, like it's actually those types of things tend not to happen. But there's always edge cases. And in particular, like the MHC portion, I think it's going to be critical that you just make sure the genotype there hasn't been recombination in the haplotypes, which are haplotypes are. Sorry, I'm using a lot of terminology here, but so like what I mean by genotype, the one worry is there's like a recombination in a protein, and it somehow generates an immunogenic like epitope that would trigger an immune response. In a very like in an interactive way that's like non-linear essentially between like two amino acids that are like aren't adjacent to each other but interact with each other, and then that gets cleaved in such a way. But I think that'll be rare from like the studies that have done this stuff, and some of like the immune matching, what they do anyways for like transplantation. For the most part, they just look at the certain regions, and they don't they're not looking for like these genotype by genotype interactions.
I see. And then one big problem that I think you mentioned on the site. So you're breeding these like skin cells together over time, and that should over time like weed out the mutations. But at the same time, aren't mutations also just like increasing over time? So doesn't that like replace that effect?
Yeah. So that's one of the things why I think what's critical is how fast our process is. Because we want to essentially minimize the number of mutations. Number of cell divisions. Because the number of mutations roughly scales with the number of cell divisions that has happened. We definitely basically want to get that down. That's like one of the levers we want to turn. Just make the process. We want to make the process as fast as possible, anyways. Mainly because then we can do more generations, and that makes everything more efficient. And so there's a natural inclination already to try to reduce that, and so that'll reduce the number of new mutations we introduce. It's also known that like in cell culture outside of the body, there's a higher de novo mutation rate in like when we culture cells in an incubator in a petri dish. So that's it's on, unfortunately right now it's on the order, like it's about an order of magnitude higher. But there are ways to reduce that actually. So we grow it in like right now we grow most people grow cells in a relatively like oxygen-rich environment compared to the body, and so what you can do is just reduce that, and that's actually known to reduce the mutation rate because you're generating less like oxygen radicals that damage the DNA. But it is just a little bit more like expensive and time consuming and hard right now.
So the number of mutations that like emerge are not related to the number of cycles, it's just related to the amount of time.
Yeah. I mean, it's related to the number of cycles too, but you essentially want to reduce the time and the number of cell divisions that the cell undergoes in your process. Yeah.
And then another aspect of this that Ivy Natal is working on is the agricultural aspect. Perhaps you could tell us about that.
Yeah. So, and since we already briefly talked about it, like the iterative portion is the key. And so,
or like, I guess what what is the goal for the agricultural part? Like, how is that going to like improve agricultural yields and stuff like that?
Yeah. So, selective breeding has been one of like humanity's like most oldest in a lot of ways, one of the more successful technologies. We've transformed plants, from barely yielding calorie inputs to like mass generating like corn. And just in the past, half century, we essentially like tripled the yield, like meat yields for certain animals. And then for like milk production, that's like doubles over the past century as well, just from like traditional selective breeding practices. And, the past decade or so, that's sort of moved on to genomic-based breeding, where they genotype the animal, and then with some degree of accuracy, they have some genetic prediction of like some important production trait. And so we essentially accelerate this by reducing the generation interval time between generations. And so that's one of the big bottlenecks animal breeding has right now, is that for, let's say, a cow, it'll take 2 to 3 years to produce a sexually mature cow that can go on and breed. And so groups have already, like the state of the art, as some, there have been ways to cut that down to like 8 months using like IVF-like approaches. And we're just taking that to sort of its logical conclusion. Instead of just moving everything into the dish and not producing any intermediate animals. And so even in like the sort of like the state of the art, they still have to produce an animal. For us, what we can potentially do is do our meiosis induction, either generate a cell fusion or an embryo, and then genotype, and then choose the ones with the highest predicted value, and then repeat that process. Take a couple more cells from the colony, and then do induced meiosis again, and then from embryo, repeat that process. And so that process greatly shortens the generation interval time. It doesn't result in like animals, like fully developed animals. So you don't have to house and install them, and it would also, you're selecting for potentially, back to your other point with selection, essentially selecting a lot more than would be feasible if you were having to produce a full animal. And so, so it just accelerates everything. So a breeding plan that would naturally take like 100 years, we can essentially compress down to something potentially like 5 years, given how fast we cut the generation interval time. And so, and it also, that opens a whole host of things where you can select on like a lot of different strategies that wouldn't be possible otherwise. Select on more traits, select on, keep genetic variants that would have otherwise been lost with like a traditional breeding one. Even in the sense that they would have been bred out. It still had a positive value for some trait, but it's just not economical to keep that in like a breeding population. But now with this process, you can essentially do a lot more complex strategies that wouldn't be feasible. This also still like applies to plants as well. Like this tech would still be valid for plants as well, actually. But obviously that's not something we're working on. But even in like fast breeding plants, not having to generate like a full plant as well as at the haploid stage, like other like there's no other complicated issues too for like gene editing, but editing at the haploid stage is kind of valuable for them as well. So the big value add is like the generation interval time, just cutting down that, and that's something that the industry is already working on, and they're actually working on them extremely aggressively.
Yeah, very impressive if it can be pulled off. I'm not sure if you mentioned this, but what role does machine learning play in your approach?
Yeah, so for us, it's essentially finding the targets to induce meiosis. It's been like the transcription factors and some of the other proteins that we think are necessary to recapitulate, not necessarily like recapitulate the natural meiosis, but like get us to the point where it would generate recombinant haploids, meaning there's a reduction in chromosome number and we have the right set of chromosomes.
I see.
Yeah.
Well, it seems like a very like potentially like like transformative technology can be applied to so many things, from reproductive issues to de-aging to like transplants without immunosuppression, the agricultural improvements. So it's very cool. I think that's all my questions. Thanks for coming on, and good luck for the work. I think it's very cool. It's very promising.
Yeah, thanks for having me. It's great to talk about some of the non-fertility related applications. I know it's like speculative and stuff, but it is in a lot of ways it's just like another potential tool and like the molecular bio tool like that people can use to help accelerate